Great. Welcome, everybody. Thank you for coming to this mentor meeting for the BioBuilder Club. Chloe's one of her most joyful and most difficult parts of her job is finding and pairing mentors with our wonderful BioBuilder Club teams. And you guys are such an important part of the program. And she really spends a lot of her talent and energy making sure that it's a good fit. We do our best. There are some real highlights, some real pitfalls that we've seen happen regularly. And so with this meeting, we're hoping to just scope out what the work ahead is and answer any questions you have, share some of what we've seen happen, and convey that we're here to help. So mostly Chloe will be driving this, but I do want to welcome and thank everybody who is a new or returning mentor to the BioBuilder Club. We have a lot of teams. I will let Chloe tell you how many, but we have the biggest, best BioBuilder Club season ahead of us, that's for sure. Chloe, did you have something you wanted to drive from?
Yes, I am sharing it right now.
Perfect.
And my computer is also having a slow day, but I promise it will show up momentarily. All right, so Laura is here. So I will tell you that yes, this is our largest BioBuilder club yet. And there's a few reasons for that, which we'll explain. And we have currently 73 teams registered throughout the US and internationally. So I wanted to just start out because we have so many new mentors to kind of explain what we're trying to do with the Bible. So our unofficial motto is all you need is a good idea and we'll teach you the engineering to bring it to life. And I think that's a really good motto for this program because it's incredibly student driven and it really, empowers students with so much agency and creativity. So we really try to support students in whatever they choose as kind of the direction that they want to go in. And we try our best to bring as authentic as possible tools and approaches to the students as well. So um, I'm sure you all know that STEM has an incredibly high attrition rate. And in a study of 24,000 high school students, they found that nearly half of ninth graders who said that they wanted to pursue a career in STEM had changed their minds just 2 years later. And this is particularly even higher rates of attrition for students from traditionally underrepresented groups in science. And they found that these changes in career and education intentions are very closely related to their high school science experiences and how those experiences shaped their self-perception and whether or not they see themselves as a science type person. So, in the BioBuilder Club, we're really emphasizing that every student belongs in STEM. And mentorship is a really vital way of demonstrating that and kind of allowing students to have not just your, expertise and talent in helping them develop a project, but also to see different career pathways into STEM. And Natalie was just saying the other day, like most students have no idea what a career in STEM really looks like. What do people in biotech actually do? And so that's really also a vital component of mentorship in the BioBuilder Club. So this year, part of the reason the BioBuilder Club is growing is because we're introducing regional hubs. So this actually started the last couple years with Georgia Tech University and their Frugal Science Academy run by Janet Sandovan. And this program has really brought in a lot of schools from the Atlanta and surrounding metro area to have students have both lab experience at Georgia Tech, summer internships, workshops, and supporting teachers. A lot of teachers, this is way outside of their comfort zone. And so new teachers especially have really benefited from kind of this networking and additional support. So this year we were fortunate to have a grant from the American Student Assistance organization. And it's really cool because it was a student-led grant-making initiative. So we got picked by students in Massachusetts. And so over the next 3 years, we're launching regional biotech hubs in Eastern, Central, and Western Massachusetts. Obviously, we have currently a large presence in Boston, with 15 of our Massachusetts teams being in the Eastern region. But we have, for the first time in BioBuilderClub, registered 4 teams in Central Massachusetts and 1 team in Western Massachusetts. So we're really trying to also have some connections between teams in that area. If you end up mentoring one of the Massachusetts teams outside of Boston, there's probably going to be additional opportunities for those students to connect with each other and with different science initiatives in the community. So heads up there. And we're growing internationally. We got to actually turn away a lot of teams this year. We have 30 teams who are not in the southeastern US or Massachusetts. So I will not read them all. This is a list of all the schools that are participating. And it's kind of exciting. This year, many schools who have participated in the past have returned with even more teams. Um, and so we have a huge variety of teams that are joining for the first time, and teams where the teachers at those schools have participated for years. And some of the projects that students are working on, this is, they're seniors now, and they joined in 9th grade. And so they've been working on these projects for 4 years, and they really get impressively sophisticated. And they're able to actually reach those build and test stages. So, and for new mentors, just an overview of the BioBuilderClub season is that it starts out with teams exploring SynBio. Now, most students really have never heard of synthetic biology. Part of it might be a naming issue, but they also really don't know really what bioengineering is. A lot of students in the pre-survey, when we ask them for an example of synthetic biology, say prosthetic legs. So there's really room there to give them a broader view of what bioengineering and synthetic biology might be. So this kind of exploration process, we provide a lot of resources and webinars and things for students to do this. And many new teams kind of explore through November and really settle on topics in December. I have started pairing mentors on a rolling basis. Some returning teams, are ready to go with their previous mentors, and some teams have come in with a very specific project in mind, and so they are off. So about 20 teams are already working with their mentors, but we have plenty of teams. So if you don't have a team yet, please do not panic. You will get one or more if you would like, and I would like that. So then teams go into the project development phase. I'm trying to be serious about deadlines this year and making sure that students are making progress on their projects, because I really don't want to be editing all of these projects the week before the final assembly. So students will be required, and I'm going to be very serious about this with the teachers, to submit kind of a design check-in in mid-January and then the very beginning of February. Well, in the BioBuilderClub handbook are examples of the templates for their posters and talks. The final assembly details are still being determined, but it will likely be March 12th. And if we're lucky, it'll be at the Reagan Institute again. And so in February, there's going to be drafts of their slides too. So there's very specific formatting requirements. And I'll also make sure that mentors receive all of that information as well, since a lot of teams do need assistance. This is their first time preparing a scientific poster. So some of the key features of the BioBuilderClub are the ways that we support students. So this is a very student-driven program, and we try to expose them early in the season while they're exploring to a lot of diverse applications of SynBio, and also to develop the kind of skills and knowledge of biodesign, how to do literature research, as well as some career talks and career exploration, and some skills like how to communicate their experiences on LinkedIn, resumes, that kind of career-oriented wraparound learning as well. So all teams have access and mentors to the full online textbook, and each team has one copy of the book that we have actually sent to them. I also wrote a BioBuilderClub handbook that summarizes the biodesign process, has a lot of linked resources and information for the presentations so that students know in advance what to expect. We did away with Google Classroom. Too many schools weren't able to access that on their school internet. And this year we're using Discord. And apparently a lot of teenagers use Discord, so that's why we went with that option. And I would love for mentors to be able to join that. The next slide has a QR code. And so on Discord, there's also within the BioBuilder server, we have a mentors channel, and sometimes teams will have very specific questions that you might not know the answer to. But with over 50 mentors, hopefully someone knows someone who can help you source that rare strain of algae. So there are several opportunities. Oh, and for teams that do build and test, and will all the teams receive this, we do provide a $100 gift card to Carolina Biological, and so teams can purchase anything they want with that. Teams are also aware that they have access to certain reagents from NEB, and so this is all in the handbook, but if they are at the build stage and they don't know how to find things, you can remind them it's in the handbook, or they can reach out to me. There's going to be several opportunities for students to showcase their work this year. Of course, we have our final assembly in person in Boston and online. There's also going to be a regional assembly at the end of the season after our final assembly at Georgia Tech. All of the projects and presentations are on our newly redone, beautiful project library. And then more and more teams each year are publishing after the end of our season in Biotech, which is a peer-reviewed journal for high school synthetic biology. It's a great experience for students to go and learn about the peer review process and practice communicating their ideas, and also giving feedback to other students. This year, from our teams last year, we had 57 teams, and 35 of them published their articles in Biotech. And that will be officially coming out, I think this week or next week. And it's free to read online. So I'll send that to mentors. A lot of your teams, if it's a returning team, might have actually published their design. And Biotech is cool because they accept a whole range of article types. So even though students might not actually get to the build test stages, they can still experience that publishing process. So this is our Discord server. I do put all of the events on Discord, so that way it's convenient to access Zoom links and things like that. You can get advice from other mentors, reach out to other teams. It is becoming more global. This year we have teams in Spain, Taiwan, Shanghai, and I think one in Indonesia. So we have a couple goals for team projects. I would say that the top goal is, oh, wait, hold on. I'll go back to the QR code. Sorry. I see cameras. And it's set up now. This link doesn't expire, but you'll probably have to put your name in, and then I have to approve you because I just had so many people. So this will be in the follow-up email as well. Just put a link that you guys can click on to. So our goals for team projects is that most teams, like the vast majority of teams, are not going to complete an entire design build test cycle. Unfortunately, that's just not really realistic in the 6-month span during an academic school year. So the teams that do actually build things are typically returning teams. And so a solid bio design is a perfectly reasonable goal. And we try to stress to students that that is a perfectly reasonable goal. Like they have not failed if they don't actually build their design. Um, and another note about this is that if they're just working on a design, it does not have to be feasible to build and test. And we note this because every year we have teams come up with very interesting project ideas that you just can't build or test in a high school lab. They are not getting access to radioactivity or pathogens or heavy metals or carbon monoxide. That's just no. But they can still make the design. So if it is a team that has the resources and will get to the build stage, keep in mind that they cannot bring heavy metals to their high school lab. I think some of the mentors on this call, including Natalie, have experienced some very interesting project ideas that were just not a good idea to build. And then I will say that even the teachers who have participated before, sometimes those teams still need support in kind of figuring out how to specify their design at the different levels of the abstraction hierarchy. We aim for projects to be at least, I say, SynBio adjacent. Every year there are teams who are just determined to work on a certain project. And at the bare minimum, it has to have some kind of living cell in it. We would like it to be a little bit more synthetic biology. But since we're trying to make sure that these students have authentic research experiences that are positive and build their science identity. If it's a little bit off topic, I'm not going to stress out about it too much. And I don't think Natalie will either. Natalie? No, she's not stressed. Okay, so most teams, we would really just definitely want them to be able to like articulate what the problem they chose is and then specify it at the level of system, device, and maybe parts. Students sometimes do have a really hard time figuring out like what cellular chassis are and chassis selection. And then it would be really cool if students understood the inputs and outputs of their system. And even if they're just working on a design, as you'll see in the handbook when it talks about presentations, they can certainly talk about what experiments they would do if they had the time and resources to get that far. So a couple of things to keep in mind is that this is not a competition. There's just a huge difference in what resources schools have access to. We have incredibly well-funded international schools with access to partnerships with biotech companies and amazing lab equipment, all the way, to very rural, small Title I schools that have no labs. And we do want everyone to have a good learning experience. And so we are really just here for progress and learning. And because the program is flexible, some teachers are actually running this in their classes. Some schools have biotech courses or they're running it as part of AP Bio or an elective. And then others, this is an after school activity. So there's definitely a lot of variation also in student commitment to this endeavor. And so my other reminder is that this is a student-driven program and all of the students are teenagers. I think that that's important to keep in mind. They do not have a lot of experience with professional communication skills or styles. They don't understand that even if they don't have an answer to the email, they cannot ignore it for a month. They really prefer texting. You might want to get someone's cell phone number. And a lot of teams are going to be like really in it at the beginning and just not cross the finish line. And other teams, you'll be like, oh my God, they're slacking off. They're never going to get anything. And then they come up with this amazing presentation. So they will surprise you. They might frustrate you. But they're learning. And so if you do run into issues, please reach out for help. I can reach all of the teachers in the program, and I'd rather know earlier so we can kind of make a plan on work on this with the students rather than, wait later in the season. Okay, so finally for my portion here, I have several teams ready for mentors that I am in the process of pairing with. So I thought I would put it out to the group if anyone has experience with structural color. It's a really cool project from a team in Shanghai. They want to make an alternative to glitter. And we've reached out to a lot of really cool biotech groups, but unfortunately, no one yet has the time available to actually mentor. So if anyone here or anyone that you know is really into structural color or sustainable glitter, please email me. Another team actually published a perspectives article in Biotreks last year about diagnosing congenital central hypoventilation. So if you're really into qPCR chips or something like that, also email me. And then I have a team that just wants to do something dermatology related. Um, so I'm not exactly sure. They're not sure yet what direction this project will go in. But if dermatology is the passion of you or someone you know, again, love to hear about it. Also, planaria. A team has decided to work on ciliopathies in planaria. And they're not really that popular of a model organism for synthetic biology. So, if you are great with planaria, I have the perfect team for you. And we still do need mentors. As I said, I think we have like 54 mentors right now. And thankfully, thank you to the mentors, all of you, for your time and talent, and also mentors who are, willing to manage two projects at once, which is quite the undertaking. So if you do know more people who might be interested, I would love for you to get them in touch. And so, here's great pictures of our students. And anyone have questions or want to ask other mentors things, advice to share? The floor is yours.
I have a question. I think I got really lucky last year. I just happened to find a group that was doing exactly what I was doing. However, I come from more of an AI background. I'm like purely computational. I have synthetic biology expertise, but I feel like I couldn't really help them much on the experimental side, which is kind of the point. So, is there like any option to maybe be like a floating AI mentor rather than like dedicated to one team where I kind of fail to help them with the crux of the problem, which is synthetic
You
biology?
didn't fail. You didn't fail.
I got lucky, but well, maybe that's because of you. You did such a good job placing. So I think
Yes, I
that
will try to
again.
place you again. I'm
Okay,
trying to pull that
perfect.
off again here.
Yeah, okay,
Um, yeah, with so many teams, we do have teams who are interested in more of the computational side and a
okay,
lot of interest recently in AI. So absolutely, I suspect that we will have another team that would be a good fit. But
okay,
thank
great.
you so much for doing that. You did not fail, trust
I
me.
actually still meet with two of them. We still meet once a week. So yeah,
Oh
they're
my God,
still
that's
working on it.
amazing.
Yeah, it's awesome. They're so great. They're the best. Yeah, it's good. Thank you.
I have a question. So thank you. I'm so glad that there's so many groups. You have a lot of stuff to to manage. But one of the things you just mentioned is that you can't bring heavy metals into a high school lab. And my group is in the build phase of a heavy metal detection
Yes, and
system.
I reached out to Ben at Arbor about that for his input because he previously worked on a heavy metal group.
Yeah.
Um, and he did say that the school did not let them bring heavy metals into the lab.
Okay. So last time we met, I gave them a task of they wanted to start with lead. And so I said, well, let's find the source of lead so that you can test. But now I feel like they cannot, if they cannot test, not sure if anybody in the mentor group, maybe this is a Discord thing, if any of you online happen to have ideas. I guess they're in Case Western Reserve. So I don't know if I don't know where that is actually, if they're
It's in
universities
Ohio, and oh
that they
wait,
can partner
there
with.
I think your wait is your team is at Western Reserve Academy in
Oh yes, maybe that's different than Case Western.
it
Sorry.
is, yes. So they're at
Ignorance
Western
here.
Reserve Academy. That's okay. It's
Okay.
in Hudson, Ohio.
Mhm.
Um,
Okay.
and that school does have a lot of resources and
Okay.
lab facilities. And so, depending on the school,
Okay.
actually, there might be some different options as far as building and testing. Now, WRA is traditionally run by Beth Pethel, but she's actually out this year on sabbatical, which I
Yeah.
didn't know high school teachers could have. And so this year, actually, it's being run by a teacher, Vivian.
Yeah.
And I don't know if you know, but she actually did the BioBuilder Club when
Okay.
she was a high school student at WRA.
That's
So
great.
I would definitely talk to her,
Okay.
because they're not necessarily like a typical situation.
Yeah.
All right. Okay. I'll reach out to her directly and ask if they have the ability to dispose of the heavy metal and like access to that. Okay.
And Natalie, I don't know if you have input for
Yes,
this.
I have at least two things that I wanted to mention. One is that it can be useful for the team to think about which section of their overall project they could carve out as a testable portion. So, when the students scope their project, they have an input and they have an output, but the idea that they could build all those complex genetic circuits, insert them into some chassis, and then just test their organism to see if it works, that's usually not a recipe for great success. It's usually a recipe for a dead organism. And so finding a way to say, well, the readout is going to be an orange color, right? Can I get that orange color generator to be responsive to something, right? Some input that is safer, right? So that is finding a way to parse the project so that a section of it is testable can be really, really useful. So that was one idea. And that's a good way to think about your project design and test as well. The second idea is that there are some teams that cannot themselves do the testing, but that partner with a local company or university or school to do the testing. I'm thinking in particular of the Andover team from last year that had been working on a plastic degradation organism for a very long time, and then they built it and they wanted to test it, and their test was using a scanning electron microscope to determine whether this organism could etch away at some plastics. That is not an instrument that high schools have, but they were able to go to UMass Medical Center or something like that to partner with a local university. So there are other ways to think about testing that may not bring the things to the school, but maybe let the school go out to the community. That would be another
And
possibility.
I will say that more and more teams are working with university labs in partnership. So that's definitely an option. And yeah, so Vivian at WRA does run that Hiroko as a part of a class. And so they have more time to kind of dedicate to that. So
Okay. All right. I'll reach out to her. Thank you.
any other questions?
Hi. Um, so I'm brand new to this, and
Welcome.
thank you. And my name is Rick, as you can see on the screen, so I'll go by that too. Um, so the microbes to replicate structural color, that is very, very, very specific. And
Yes, it is.
um, I think I know enough to understand what it means, but I mean, um, and I'm not sure if there is no one here that actually is an expert in that kind of thing. What level of expertise are you looking for to fill that role?
So it really okay, so it really depends on the school's situation. Because this is at an international school in Shanghai, they do have access to more resources than the typical high school does. And so I was just wondering if anyone had structural color experience, and it sounds like maybe if I can't find anyone, I will reach out to you, Rick. But in this case, because they're interested in carrying out very specific experiments, I have been reaching out to people who publish specifically in microbes to replicate structural color. And so hopefully that will be an option. And then being in Shanghai and speaking Chinese is also good because there are more resources from labs in China that might, be able to help them with that.
Okay. Thank
But I
you.
was just wondering. So if anyone for any of these topics, if you know anyone or you're willing to take a stab at it, let me know. But yeah, so like the mentor pairing process is kind of complicated because it really does depend on the context of what school it's at.
That
But I
makes
appreciate
sense.
them.
Okay, very good. Thank
And
you.
for the new mentors on the line, I'll just zoom out just a smidge and say you have so much on the ground expertise already that the students really aren't they are not where you are. And so it can get very in the weeds around their project, but it is also really valuable for them to think about, what is the control you would want in that experiment? How will you measure that? How will you know? How will you report on that? Is that safe? How will you get that? How do you know it's true? Is there another paper for that? How do you read this paper? What does that graph show you? So it is, the students are very driven by their project, but a lot of what you bring is the process of science and the understanding of how to conduct meaningful investigations, which I think students generally don't have an opportunity to do in high school. That is, a very open-ended blue sky idea that they need to get really granular on. Some of it is, how do I find a structural protein that will generate a color? But also, how am I going to measure that? How am I going to, source that plasmid? How am I going to control the expression? And a lot of that is fundamental science, fundamental gene expression, and your understanding of how you became scientists and your growth as individuals who went into this process of discovery and experimentation. So I'll just
We're really using like these very specific topics that the students need as like secret vehicles to teach them how to be scientists. So the goal isn't necessarily to actually learn a ton about structural color, but like Natalie was saying, that research process. And so even if you're not, the world's expert on this topic, they probably in most cases don't need the world's expert on that topic.
Thank you.
And I also did ask mentors on the mentor survey if you are comfortable in mentoring a team through a build project, because we do know in advance what teams are likely to build their project versus design it. And so, like even Calista was saying, if you don't have a lot of like experimental background, that's really probably perfect still for a design team that just really needs to understand that process and how to find information. It's very difficult. Students probably haven't done literature research before. This might be their very first time reading a scientific article. Usually for the final assembly, writing their abstracts for that is the very first time they've written an abstract.
Hi, I'm also new. My name is Ilene. Nice to meet y'all. I had well, I just wanted to mention I also have I don't know if it is exactly the same, but some experience doing, fractal patterns in bacterial colonies that could be potentially applicable to that group as well. I don't know if it is exactly what they are thinking about, but it could be something. I don't know if I can. I was actually gonna well, I can share some of the details later with you. And then for the
Yeah.
other question that I had was for the dermatology project. Can you give us a little bit more detail on that one? I work, for example, with some students that do genetic engineering in C. acnes. So that could potentially be relevant, but it could be that they are thinking about something completely different, like, for example, metabolic engineering to produce a compound that you can use in like a lotion or something. And if that's the case, um, it's a little different to what I do in that realm.
That's a really good question. So the dermatology team is a new team, and sometimes new teams come in and they're just interested in a certain area and they don't really even have necessarily the research skills to narrow it down to a specific project idea. And so I love being able to pair teams like that with someone like yourself who has experience in dermatology, and you can also kind of, present your ideas for projects and that. Yeah, Natalie.
Sorry, I was
Oh.
saying goodbye to Kise.
Oh, you're waving.
Yeah, thank you.
Sorry.
Thank you for being here. That's all good. But while I'm jumping in, I'll say what Chloe is saying is exactly right. You can, there's always a little bit of a tension. This is a student-driven project, but you can guide them towards an endpoint that you think will lead to greater success than another, right? Like if you have a team that wants to turn carbon monoxide from a smokestack into diamonds, that's a very cool idea, but maybe figuring out how to harness the effluent from a smokestack might be the first step. So you can move them so on a path that you're there as a guide. You can certainly influence, you don't control them. They're teenagers. It's impossible, but you can guide and influence their thinking for sure. Yeah.
Okay, sounds good. Yeah, I'll probably reach out about those two projects and then you
That would be
guys can
awesome.
determine
Yeah.
whether if my expertise is useful for those or it can be for something else. I don't know.
I really appreciate that. Thank you. Yeah.
Hi, I'm Aditya. I'm very new to this group. But as Alisa mentioned initially, like a floating mentorship, you were talking about dermatology. I don't have any expertise with dermatology, but I do make sunscreens from cyanobacteria at this particular. So I may have something to share which dermatology people can test.
That would be
At this point,
wonderful.
yeah. So at this particular point, I cannot perform certain dermatology related work at my laboratory because it needs number of yeses from the university and from the council and so many things. But if somebody already has them, so we can have like a quick test and that would be very helpful. So I don't know whether that will contribute as a floating mentor, but that can be one of the contributions from my side. But yeah, please let me know if that thing works out like that.
Thank you so much.
Yeah.
We have such a great group of mentors, and I just really want to convey like how appreciative we are at BioBuilder that you're doing this for these students. And like Natalie was saying, what you know about science is so important for these students to see and experience. And none of our mentors fail. Like this isn't a process that you can fail at.
That is exactly right. I mean, how amazing that Alyssa's still conversing with her team from last year because they're just so into it, and how amazing that a former BioBuilder Club student is now leading a team as a teacher at a school, right? And
She's leading 9 teams.
nine. All right. Well, so take a big swing. Yeah, it really is. It is incredibly impactful. It is essential for making this program run as it grows, obviously, but that's an us thing. But the work you do with the students is tremendous. It really, for many of them, is the first time that they've had a chance to really engage around scientific ideas with a practicing scientist. And that is really life-changing for them. It lets them embody that sense of a community of science that is so important to the advancement of science. And
Well, that is all I have.
3 cheers for Chloe. This is a program that she loves, and she is
Love this program.
rocking it. As I say, she is a victim of her own success here with 73 teams now, but we will make it work. We will make it work.
Well, thank you, everybody. I'll send this information in a follow-up email. And if you are one of the mentors who had some expertise or ideas for these teams, I would love to hear from you.
Yes, thank you.
Thank you, everyone.
Thank you all. Great
Thank
to
you,
see
Chloe.
you.
Thank you, Natalie.
Bye.
Good to meet you.
Bye.
Chloe, that was great.
That was good. Okay.
Okay,
Excellent. Thank you.
bye.
Bye.